Good. So in January, the session 2 is about gut microbiota health axis. So this particular series, the main title is Gut Microbiota, a Healthy Partnership. So we'll be talking about bacteria in the gut, which are beneficial and also essential, not only beneficial, they are essential for our health. When we talk about gut, we're talking about the gastrointestinal tract, which starts as the upper part, as the oral cavity, goes down to the stomach. From the stomach, it feeds to the small intestines, which is the coiled light pink section in this image. And then there is the ascending colon, transverse, because it moves from one to the other side, and then the descending into the rectum and the anus. Okay, so this is the first overview. There's a physiology of all this, so you can go through this through the YouTube video. And we also said that the main function of the gastrointestinal tract is actually to digest food and to absorb the nutrients. That's not the only function. It has multiple functions. And one interesting function that we also will allude today in terms of health prevention is actually it's an immune learning hub. So the immune cells, which are important for our immunity, are learning about the environment. And therefore, why the gut? Well, the gut, as you can see from the oral cavity to the anus, is actually an external tube inside us. So it's all external. Food comes in, digested, and this waste is removed, excreted. Okay? So in the gut, there are immune cells that learn from this environment about possible future infections. And therefore, it's very important that these immune cells, whilst we're learning, so they have to be activated, there's also a very important mechanism, which is called tolerance, which means there should not be an acute immune response. Okay? So it's only learning. Okay? And that is called tolerance. And we also will discuss a little bit about that today. But in addition to the immune learning hub, there is also the production of neurotransmitters and also activation of hormones. And there is the microbiota. Microbiota are these bacteria, viruses, and we're going to focus only on bacteria. And these bacteria are essential and beneficial because they produce substances that are beneficial and health promoting. Okay, and these substances that they produce they are called metabolites. Well, they are the output of metabolism. We know what metabolism is, and whatever the metabolism produces, we call those metabolites. Okay, so if you see the full sort of function image here, the gut and the associated microbiota are important for digestion, absorption, and metabolism. And also important for gut, brain crosstalk, so important for mood, satiety, and pain, and okay, so the neurotransmission part, and also important for immune function, both as defense, as the learning hub, and also in terms of tolerance, which is extremely important in allergy. Good. So we also showed the microbiome in the overview, so it's already in that YouTube video. And the microbiome is the community of microorganisms, including bacteria and viruses, as I said, and they are extremely important for health promotion. And in fact, we're very well known that the wider the diversity of this gut microbiota, we call them, the microbiota in the gut, the healthier the composition. We know a lot about these microbiota because in 2008, there is the establishment of the Human Microbiome Project, and a lot of samples have been analyzed. Okay, and this microbiota are definitely affected by various things, included diet and lifestyle, and also age is associated with reduced microbial diversity. So if you look at the gastrointestinal tract, there's the upper and the lower. Okay, going from the upper to the lower intestinal tract, you see that the number of microbiota present increases substantially, with the highest amount in the colon. And basically, this is the main source of reading the microbiota in the laboratories, because we take a fecal sample that represents the microbiota in the colon. And as we can see, as we will see soon, that the microbiota in the colon are extremely important in producing the health-promoting metabolites. Now, even the diversity and the type of bacteria between the different areas is different because of the different oxygen levels that there are in the various areas, and also because of the pH, the acidity. Okay, so this is the only slide where I actually have a list of these important bacteria. So it's not important to, of course, go through all these, but I wanted to show you that there are like the yellow box here, which is represented by the top right small pink box. These are the firmicutes. There are 200 different groups of these, which we call genera, and these are under the genus name. And although we don't need to know them all, of course, when we read what's present in the stool sample, that gives us a lot of information of what type of restoration one would need to do to regain the balance, because the most important is having the diversity, the right abundance, and the right balance of these bacteria to remain healthy. So that's what it is, actually, gut health. Okay. We will mention today Faecalibacterium prausnitzi, which as from now on, I will just say F. prausnitzi. We'll mention today Akkermansia municiphilia, and I will just mention Akkermansia in this case. And we will mention Bifidobacterium in the blue box. And this is one of the bacteria that most probably you know about, because it's used extensively in functional foods, in yogurts mainly, and this is the Bifidobacterium bifidum, which is very beneficial. Okay? But the main types of bacteria in our gut are the yellow box, as I said, represented by the pink top right box, and the green box, which is represented by the blue-green box on the top right. Okay, and in fact, when we look at this list of bacteria and we read them and do the abundance of these bacteria, we will see that in a healthy individual, the distribution, if you look on your left, right, there is the pie charts which show the colors. Now the pink color represents the top right pink, all right, and also the green blue. Okay, those are the main ones. Okay, and in a healthy individual, this is the particular ratio that we expect. But of course, we also need as I said, like the bifidobacteria, which are a small portion, but they are also extremely important. They should be there. Okay? And then when we compare these healthy pie chart with the obese pie chart, of course, we immediately see that there is a red part, which is very low in the healthy in case of obese, or in case of obesity. And that red fraction represents this box here. These are the proteobacteria, and these are actually activating inflammation. So they stimulate inflammation, and they are what we call pathogenic. So they are harmful bacteria. Okay, and we call that pro-inflammatory. Okay, and we know of these bacteria when these bacteria take over the balance. Okay, so they will increase and at the detriment of the beneficial ones. Okay, then you would have disease. So inflammatory bowel disease, you can actually see that like Crohn's and ulcerative colitis is a result of increased amounts of these proteobacteria, cystic fibrosis, and also the very well-known Helicobacter pylori which is associated with stomach cancer. So if we start comparing individuals, then from these abundances and diversity, we will have an understanding of the condition in terms of the balance in gut microbiota, and also relate that to specific symptoms or diseases. Now, the important thing is that, of course, to prevent, we need to identify this before the disease, and therefore it's important that one understands if there is any differences, okay, which can be restored before disease will start. Okay? Now, this particular balance, there are specific factors that can cause the imbalance, and this, as I mentioned, age, but diet is really affecting this, stress, medications, and also the presence of pathogenic microbes. So in terms of the terminology, the imbalance of gut microbiota is called dysbiosis, and dysbiosis is associated with various conditions and also diseases, and it is associated with weight gain, obesity, insulin resistance, leaky gut, inflammatory bowel disease, and also depression. So from all this information that we gather from a fecal sample, and we look at the abundance and diversity of these microbiota, then we actually set up an index. And there are sort of three main parts of this index. If it is 1 and 2, it's non-dysbiotic, so it is in balance. If it is 3, there is mild dysbiosis, and if it's 4 and 5, it's severely dysbiotic. Now, if we take clinical studies within even healthy populations, we will find that 16% of individuals have actually a mild dysbiosis, and this is fully restored through specific prebiotics and programs and diet plans. Then we have individuals with irritable bowel syndrome. This is actually the syndrome that we will discuss today in terms of microbiota and health. And irritable bowel syndrome, IBS, 70 to 80% have actually a profile which is higher than 3. Now, ideally, to prevent, it is in the region of 3, so that you can actually restore it quickly. But you do it in an sort of action according to the dysbiosis, so according to measuring the difference and the discrepancy. In terms of inflammatory bowel disease, now we're moving into the diseases, and that's 4 or 5, an index of 4 or 5. And at that point, the restoration of dysbiosis is extremely important, but of course the disease has also to be treated. And the way that this index is sorted and analyzed is by looking at all these bacteria, but putting them into functional profiles. We would like to see, and we will see these, mention these in some examples soon. Butyrate is a very important health-promoting metabolite, and therefore these producers of butyrate should be present. If they are low, that of course is not correct and needs to be adjusted and restored. We look at those bacteria that are important in setting the mucosa, the mucus. Again, in the overview through the physiology, I mentioned that there's a mucus layer above this one-cell thick gut lining. Okay, if those bacteria that maintain this mucosa, this mucus, then if they are low, then that is also needs to be restored. We always look at this very specific bacterium, which is F. prausnitzii. That is extremely important because it is the main producer of butyrate and also a number of additional health-promoting substances. Now, we also look at other functions like gut barrier and for leaky gut. So we will have specific bacteria, which we understand that there is a leaky gut, for instance. And also we look for those bacteria that are pro-inflammatory. Basically, we're talking about sort of the red portion of the pie chart. So if we start taking into account the microbiota in terms of function, we look at those that produce health-promoting metabolites. Now, this is a new pie chart here, which shows the importance of specific metabolites that need to be produced daily. Okay, so this is a continuous thing. So the balance is extremely important to maintain these health-promoting substances. So I already defined metabolites, which are the products of some kind of enzymatic process. An enzyme is an active protein that change a compound into a different compound. So it is an enzymatic process. And these enzymes have to be present, okay? And although the major enzymes are produced by the human body, and actually most of them will come from the pancreas towards the gastrointestinal tract, or from the liver via the bile, so sort of the production of bile acids. All these are important, but there is a multitude of enzymes that are only produced by microbiota, by these bacteria. And in fact, that repertoire of enzyme is much higher than what we actually provide to digest the food ourselves in terms of human cells and human tissues. So having the right balance is extremely important because it will change the metabolism of the person. And that's why a disbalance would eventually result in what we call the metabolic syndrome, okay? Because the metabolites are changing and the inflammation will increase. So there are various ones, and we go one at a time. Okay, we will start with the purple one on the right. Okay, those are the bile acid metabolism. It's important that the prime bile acids are changed into secondary bile acids by specific bacteria. So let's start with that. There are various metabolites that you saw in that pie chart. So there are the bile acids, there is the SCFAs, these are the short chain fatty acids. There are vitamins, there are specific amino acids that are metabolized, and you require specific bacteria. There are neurotransmitters, as I mentioned before, and that is, all this is altered by dietary intake. And that plays a role in the metabolism of the organism, of the human being in this case. Okay? And this balance will provide us and distinguish a normal healthy gut from a dysfunctional unhealthy gut. And to try and explain to you the relation between gut microbiota and health, I decided to give you an example of what we call the functional gut disorders. We call them disorders because it's still not a disease, okay? FGDs, and this is exemplified by irritable bowel syndrome, which I will refer to as IBS. Okay, so let's go into functional gut disorders, and I will only tackle IBS. So if you look at the bottom part, this really says it all. If you have the bad gut microbes, this will increase the inflammation, and that is equal to poor quality of life. And that's because these bad gut microbes will affect immunity, will affect production of vitamins, will affect totally the metabolism. Because all this is affected, then there you have an increase in inflammation. And this is all related to metabolic syndromes such as obesity. Now, there's a complex interaction between food, ourselves, the host, and the microbial metabolites. And all this complexity and interaction will eventually result in a specific functional gut disorder if there is a disbalance. Okay, so it is not simple, but I will take you through step by step the disbalances that we know of. So if we take the irritable bowel syndromes, IBS, okay, it's known, it's reported that 11% of the population is diagnosed worldwide with IBS. It is important to understand the cause, and that's why today the measurement of gut microbiota became extremely important in the preventive side. But in terms of IBS, it also is important because you can restore this. There is an intervention, a possible action, okay, using diet and nutrition and prebiotics and potentially sometimes probiotics, depending on the disbalance, and people can actually find a solution. So understanding the cause is definitely crucial to do the right intervention. So if we look at this pie chart sort of slowly, there's the bile acids, and it's well known that in IBS are characterized by reduced concentration of two specific microbiota, two specific bacteria, the bifidobacterium and the Clostridium leptum. And these two bacteria are important because they have a specific enzyme, which the human cells do not, to transform the primary bile acids into secondary bile acids. Therefore, if you have lack of secondary bile acids, that would result eventually in disease. Then if you look at the green part, which is the short chain fatty acids, these are composed of they produce acetate, propionate, and butyrate. Okay, these are the primary ones. And 80 to 90% of these short-chain fatty acids are actually used by our body. Okay, so they are extremely important. Now, the source of that could be complex carbohydrates like dietary fibers, but also the mucus layer of the gut lining. There are specific bacteria which are extremely important to measure and to restore, that actually feed on mucin, which is a protein in the mucus, and produce acetate. That acetate eventually becomes the food of other bacteria which produce butyrate. So this has become a chain reaction. So it's important to look at the whole thing and start acting accordingly. So if there is low short-chain fatty acids, such as in the case of IBS, and that would result in a very low intestinal barrier integrity. As I said in the first session, the gut lining is only one cell thick, and therefore it's important that it is tightly together. If you lose that strength in the cells, and there is other chemical and metabolites that are important in keeping this tight array of cells, that means that means it is barrier integrity. So you can lose it if you don't have the short-chain fatty acids, since short-chain fatty acids are actually energy source for the cells of this gut lining, which we are calling enterocytes or epithelium. Now, the main producer of butyrate is F-prosnitz, but of course there are others which I have listed there. So F-prosnitz make up 5 to 15%, depending on the individual. You should not have less than that. It's turn metabolize undigested dietary fibers into butyrate, and also have a number of anti-inflammatory properties, and that maintains the gut lining, as we just said. Now, another extremely important bacterium, which needs to be measured to look at any dysbiosis, is Akkermansia mucinophilia. Mucinophilia means that it loves the mucus, okay? So it actually is the green bacteria that you can see on your right dark blue background side. Okay, so those green bacteria are actually Akkermansia, and they actually feed on a protein in the mucus and stimulate cells that produce the mucus to produce more. And that's important because they replenish mucus, and the mucus layer is always thick. But they have other properties. Okay, they also are important in maintaining the tight junctions. These are the blue arrows tightly together, which when you look at onto the comparison of the left figure, then you can see that they are not tight anymore, and therefore the cell junctions are now open. So this is all important and related to Akkermansia presence, which is around 4% of the microbiota in our gut. Okay? Now, if this particular bacterium is very low, then there is no mucus, then there is the integrity is compromised, and therefore there is entry of specific substances produced by bad bacteria, which would result in inflammation, but also eventually diabetes, insulin resistance, and also fatty liver. So there are various complications because you disrupt this balance of microbiota, and in terms of Akkermansia, this is extremely important. Now, another point which one would need to specify, if you, for instance, take probiotics from a pharmacy with the various types of bacteria, Akkermansia is not there. So Akkermansia is not available as a probiotic, and because it's still not safe to provide as a probiotic. So there are various other ways how to increase the Akkermansia amount if somebody has low levels. And studies have shown, for instance, that Akkermansia increase is possible through the supplementation of omega-3 rich oils and polyphenols. Okay, so this is one way. There are other ways, but definitely that is the best way to start having Akkermansia in balance again. And this is why this is actually showing this. So on your right-hand side, this is normal balance, which we call eubiosis. You don't want to lose that. It's the right balance of firmicutes to bacteroidetes, and also right before the bacterium is high. This provides enough butyrate and enough anti-inflammatory substance to maintain an anti-inflammatory capacity of the tissues. If you lose this balance, of course, this biosis kicks in, which is the red part, the red side on your left, and that results in inflammation with all the causes that we mentioned before. Okay? Now, moving from dysbiosis to eubiosis, the normal balance, okay, there is the importance of omega-3 rich supplementation, which is important in case of dysbiosis. Now, in addition to these health-promoting metabolites, there's also other bioactive compounds which are produced by the bacteria in our gut. One of them is neurotransmitters. 95% of 5-HT, which is the serotonin part, which we call the happy hormone, okay, is produced by the gut epithelium, so the gut lining, specific cells, of course. Dopamine is also produced from the gut lining, which is linked to depression and anxiety, and it's known to be very low in IBS individuals. And also GABA, which has an anti-inflammatory effect, is also reduced in IBS individuals when compared to healthy control. So all this is mainly because of the dysbiosis that exists, which then disbalances everything. Okay? Now, another point about bioactive compounds is that there are also hormones that are activated within the gut. And if you go on your top left, there are various organs and tissues that produce estrogens that will actually run in the bloodstream. Okay, but estrogens, of course, to regulate the amount of estrogen in the bloodstream, There is they are what we call conjugated, but basically what it means is that a protein is bound to the estrogen, and that signals the bound estrogens, which we call conjugated estrogens, to be excreted via the urinary tract or the fecal excretion through the intestinal tract. What's important here is that within the intestinal tract, there are actually specific bacteria that would remove this protein from the estrogens and recirculate the estrogens towards the bloodstream. So there is a huge regulation of estrogen levels in the bloodstream. And again, we have the microbiota bacteria in the gut here, which play an important role in the regulation of such an important hormone. There are other bioactive compounds, which are basically in this figure. We talked about the short-chain fatty acids, which are the green boxes, which of course are very important, lower inflammation, increased barrier integrity, so everything is health promoting with these. Now, branch-chain amino acids are also important, but high levels of these would result in sort of increase in visceral fat, insulin resistance, and other complications. TMAO is also a product of bacteria through the liver. Then eventually good levels are beneficial, but very high levels will become pro-inflammatory and increases the risk of cardiovascular disease. The metabolites of gut bacteria are also involved in inflammatory modulation, so modulation of the inflammation process, okay, and also of immunity. And in fact, here I'm showing actually the control in terms of food allergy. Okay, there are a number of bacteria that are important, but here I have to mention one. So this is exemplified by a bacterium called B. fragilis in the GI tract, and they produce this polysaccharide A. And this polysaccharide A is very important to activate specific immune cells. So immune cells are made up of T cells and B cells. So this is a special type of T cells, and actually it's called Treg. It's called Treg because it regulates the immunity. It's a T cell which regulates the immunity. Actually, it inhibits an active immune response if it is stimulated with IL-10. In terms of food allergy, this totally changes. Tregs will totally change because there is no stimulation of specific molecules within the Tregs. They change and they actually start activating the T cells, the immune response. And you don't want to have that because that would result in food allergy. So dysbiosis itself also results in food allergy through dysregulation of these very important T cell regulators, which are dependent on the presence of specific bacteria exemplified by B. fragilis. There's also amino acid degradation, and here I just mentioned tryptophan. Tryptophan, in the presence of specific bacteria, is metabolized into various health-promoting molecules. One of them is 5-HT, which is serotonin. That is extremely important in the gut because the gut is autonomous in terms of nervous system, so it's not controlled by the brain. Of course, digestion is continuous all the time, unless there are specific hormones that stop it, of course. But the motility of the gut is dependent on serotonin and other neurotransmitters that are produced by the gut itself. But there are other important compounds like IPA here, which are important in tight junctions to maintain the integrity of the barrier. So tryptophan is extremely important in the homeostasis, in the regulation and the balance of this cell barrier, and also in the motility. But of course, because it produces 90% of serotonin, is also important systemically in terms of mood. Okay, so for instance, in the overview, I told you if you're not feeling well today, you know, you're feeling down, take a tryptophan shot, and that's a nice almond milk with dark chocolate. Yeah, so a good hot chocolate. That's a good tryptophan shot for you. Some bacteria actually produce vitamins, and some vitamins are only produced in the presence of specific bacteria. Human cells are not available. I cannot produce these vitamins. And in fact, about half of the microbiota genes actually have these enzymes that synthesize 8 key vitamins. Not going to mention them all, but like biotin, you know, folate, niacin, most probably, thiamine, riboflavin. So these are the really important vitamins, and versus, and also vitamin K2. So vitamin K2 is only produced through specific microbiota. Okay, so human cells are not cannot produce it. Okay. Now in IBS we find low vitamin B6, for instance. Okay, and that's linked to inflammatory conditions. Some note of interest here is that you remember you said the yellow box, the pink pie chart, yeah, the pink and the other part. So in general, the pink are very high, but in IBS it is even higher. So there is like the firmicutes take over most of the pie chart, and this actually this particular firmicutes, this pinkish part of the pie chart, do not actually synthesize enzymes that produce vitamins. So definitely IBS individuals are vitamin deficient if they don't take supplements. So I said as well that there is a lot of interaction between diet, these metabolites that we just mentioned, and physiology and our own physiology. And if there is disbalance, this interaction, okay, would result in eventually disease. Okay, so irritable bowel syndrome is a condition which can be restored. But if maintained, it definitely would result eventually into an inflammatory disease. Once it's an inflammatory disease, like inflammatory bowel syndrome, and cardiovascular diseases, and so then it's very difficult to restore. Although it's important to restore at least the disbalance in terms as a combinatory way, sort of combinatory to treatment, of course, because dysbiosis needs to be restored in these patients as well. So this is just one figure showing the interactions. So we talked about the yellow box butyrate being low, which would result in dysbiosis and permeability of the cell membrane, permeability of the cell lining, okay, which would result eventually in inflammation. So we showed it in a different way, but this is the lower part of the figure. But then the upper part, if there is high consumption of food that has a lot of choline and L-carnitine, that would result in very high amounts of TMAO. High amounts of TMAO would result in these foam cell formation in the vessels, blood vessels, which is called atherosclerosis. Therefore, the combination of dysbiosis due to butyrate, permeability, the presence of inflammation, the presence of atherosclerosis, altogether can lead to heart failure. So this is the interaction between all, and it all comes from dysbiotic condition as a cause of inflammation, and also through bacteria that produce specific compounds that are beneficial if they are in the right level, but they become harmful if they are at high level. Hence, we always state it's not the type, it's the diversity and abundance and the balance of these bacteria. Okay, so it's not only the type. So yes, there are gut restore programs. So you have to restore the lipid profile to have healthy cells. You have to make sure that your vitamin D3, K2 levels are optimal. you have to measure, one have to measure the dysbiosis, adjust the dietary intake, so there should be good plans, and the plans should include prebiotics and probiotics depending on the bacteria. So I would like to thank you, and please register so that we can send you information about the next session that will be in February. And the topic of that session in February is going to be gut microbiota, diet, and cholesterol.